All right, so I am going to start labeling the skeletal muscles, the major 24 skeletal muscles that I've asked you to memorize. I'm going to start with number 21 or number 1, excuse me. So number 1 is the frontalis. Number 2 is the orbicularis oculi. Number 3 is the temporalis. Number 4 is the masseter. Number 5 is the orbicularis oris. Going to number 6. Number 6 is the deltoid muscle. Number 7 is the pectoralis major muscle. Number 8 is the biceps brachii. Number 9 is the rectus abdominis. 10 is the one that comes across like a little band right here. That's the sartorius. 11 is the center of the thigh. That's the rectus femoris. 12 is the medial muscle here in the thigh, which is the vastus medialis. 13 is kind of in the middle of your calf here, the sole. That is the soleus. 14 is over here on the lateral side. That's the vastus lateralis. 15, excuse me, is the tibialis anterior. Over here, we have 16 is the occipitalis in the back on the occipital bone. 17 is the trapezius. 18 is the triceps brachii. 19 is the latissimus dorsi. 20 is the smaller butt muscle, your gluteus medius. 21, gluteus maximus. 22 is the biceps femoris. And then 23 is the gastrocnemius. And then 24 over here is referencing the brachioradialis. So in this section, we are going to be drawing a skeletal muscle, which you have done several times, and this should be kind of a cakewalk for you. But here's the bone, and then we have tendon that would be connected to bone. And then if we draw the cross section of skeletal muscle out like this, and then that's a circle, and then we see other circles inside of this cross section, which are bundles of skeletal muscles, and those bundles are called fascicles. So let's bring out one bundle. So that would be a fascicle. And then inside of there, let's do them in blue. We have more circles. And those are the individual skeletal muscle cells. Pull out one of those. So here we go. So starting from the bone, we have out here, we have the bone, then we have the tendon, then we have the entire skeletal muscle, which is covered by the epimysium. We cut the skeletal muscle in half, and we see these fascicles. The fascicles are covered by the paramysium. The paramysium covers the fascicle. The fascicle is a bunch of muscle cells or fibers, and I've drawn one in blue, and they are covered by the connective tissue sheath, the endomysium. This is a drawing of a sarcomere. So the sarcomere is the contractile unit of skeletal muscle, and it runs from Z-disc to Z-disc. And there would be another sarcomere over here and another sarcomere over here. And they all are part of, I'm going to do this circle here, they make up a myofibril. So this would be a myofibril. And then you have another myofibril down here. That is the cytoskeleton of a skeletal muscle cell. And then myofibrils are made up of myofilaments, which I'm going to draw next. And the myofilaments are the actin and myosin. So the thin filament that is anchored to the Z-disc, so you would have one going this way, and you would have some going this way for the sarcomere over here. This is actin. thin filament. Let me continue to draw my actin. So remember, the actin does the actin. It does the movement. And then in the middle, we have the thick filament, or the mighty myosin, like this. You might see a little bit of myosin over here. We'll just say that's kind of off the page because those are neighboring sarcomeres. The center, I'll draw this in red here. The center of a sarcomere is the M line, okay? This area where there is no actin present is called the H zone. This area right here, where there's only actin and it looks lighter, is called the I band. This area, which includes maybe some actin and some myosin, is the A band here. I think I misspoke over here. This area is the I band, the light band, because it only has actin. And then over here, you have another I band. So it's the light band, because if you look at the striations, this is the part that looks lighter because it only has actin, the thin filament. And if you remember, during the sliding muscle filament theory, these mighty myosin heads come out and grab on to the actin and pull them towards the M line, causing the H zone to disappear. So in this sheet right here, I gave you a drawing in class, and I'm just going to reference that. There's 23 different things, and I'm just going to change something very quickly. This should say voltage gated potassium channel. So this should be a voltage gated potassium channel, not ligand. All right, we have these four molecules that are important for muscle cell contraction. So we'll start with calcium. So what does calcium do? Well, calcium is located in two places. Calcium is located in the synaptic cleft, and as the action potential propagates down the axon and then ends up in the axon terminal, voltage-gated calcium channels are opened in the axon that cause calcium to come into the axon terminal, which causes the neurotransmitter to be released. And in this case, we're talking about acetylcholine. Calcium is also inside of an actual muscle cell, stored in the sarcoplasmic reticulum. And when the action potential is propagated across the length of the sarcolemma, or the plasma membrane of a muscle cell, voltage-gated calcium channels are opened in the sarcoplasmic reticulum, and calcium will end up being released to the sarcoplasm, so inside of the muscle cell, so that it can go bind with actin to cause the sliding muscle filament theory. ATP is responsible for fueling myosin to be able to be ready to grab onto actin after calcium binds to it and takes away that troponin and tropomyosin to be able to actually pull actin towards the M line. Sodium ions, so they are going to be brought into the cell after acetylcholine binds to the ligand-gated sodium channels. Those sodium channels will open right up, and now sodium, which exists in a really high concentration outside of the cell, can rush in, and that will end up causing depolarization of the individual muscle cell. Potassium ions, which exist in a really high concentration on the inside of the cell, are responsible for repolarizing. So after we've gone from resting membrane potential of -70 to +30, and then we hypodepolarize, to like +40, +45, what's going to happen is that the sodium channels will open, and the voltage-gated potassium channels will open, and potassium will rush out of the cell, causing the cell to repolarize so that it can receive another stimulus from the nervous system.